You know, in the microscopic world, some of the most dangerous things are literally designed to be hidden. Bacteria have these incredible defense mechanisms, and one of the best is basically an invisibility cloak. So how on earth do scientists see something that's meant to stay unseen? Well, let's dive into the super clever technique they developed to unmask these hidden threats.
So that's the big problem, right? We're talking about the bacterial capsule. It's this protective, kind of slimy layer that surrounds certain bacteria. Not only does it protect them from our own immune system, but get this, it also protects them from the very tools we use to see them under a microscope.
So what is this cloak even made of? It's a layer of polysaccharides, which are just long chains of sugar molecules. But the really key property here is that it's non-ionic. In simple terms, that means the capsule has no electrical charge. So the colorful dyes that scientists usually use to stain cells, they're just repelled. They literally slide right off.
Okay, so if you can't stain the capsule itself, what do? Give up? No way. This is where microbiologists got really clever. Instead of trying to fight the capsule's properties, they came up with a brilliant workaround.
It all boils down to two totally different ways of thinking. A normal positive stain tries to color the cell, right? But that non-ionic capsule just says, nope, and stays clear. The solution is a negative stain, which does the complete opposite. It stains the background, everything around the bacteria, and that reveals the cell in its capsule, kind of like a silhouette against a bright light.
It's such a simple but brilliant flip in thinking. It's kind of like trying to see the shape of your hand in the dark. Instead of trying to light up your hand, you turn on a light behind it. By coloring the whole environment, that invisible capsule just suddenly pops right into view.
Okay, so we've got the why. Now let's get our hands dirty, so to speak, and look at the how. How do scientists actually put this theory into practice and get from a sample to that amazing image?
The process uses two main players. First, you've got a negative stain, like India ink. It's a dark, acidic stain that's going to color the background. And second, you have a positive counter stain, like crystal violet. That one comes in later to color the actual bacterial cell that's tucked away inside the capsule. The procedure itself is pretty straightforward. You mix your bacteria into the India ink and spread it nice and thin on a slide. Then, and this is the most important part, you just let it air dry. You absolutely cannot use heat to fix the sample. Why? Because that intense heat would just completely destroy the delicate capsule we're trying to see. Once it's dry, you add the crystal violet, which can sneak through the capsule and stain the cell body purple.
All right, so we've done all the work. What do we actually see at the end? And maybe more importantly, why does this whole thing matter so much? This is where the technique goes from a cool lab trick to a vital tool in medicine.
The final result is, well, it's just beautiful and so incredibly clear. Under the microscope, you see this dark, stained background. Then you see the small, purple-stained bacterial cell. And right around that cell is a perfect, clear, unstained ring or halo. That halo? That's the capsule.
And seeing that halo is a really big deal. It lets doctors identify dangerous bacteria like Streptococcus pneumonia, which is a major cause of things like pneumonia and meningitis. The presence of a capsule is a huge red flag. It means the bacterium is way more dangerous. So this makes capsule staining an absolutely essential diagnostic tool that even helps us develop new vaccines.